Right, anyway. Right, B2, cells and control. This is what you need to know. So mitosis is the first thing we're going to look at. And mitosis makes identical cells. So one cell splits into two identical cells. Another thing that you need to know is that the cells are diploid at the start. They have pairs of chromosomes, so 23 pairs of chromosomes. And it makes two diploid cells that also have 23 pairs of chromosomes. So interphase is when cells are doing their normal job, but interphase also is where the DNA replicates. So you get 23 pairs of chromosomes in a cell, but that makes 23 double pairs of chromosomes. So here you can see on the left, there's 2 copies of the red chromosome, 2 copies of the blue chromosome. But here it makes these kind of double copies. And then what happens after mitosis is it splits and it makes exactly what you had at the start. So mitosis makes 2 copies of diploid cells, makes identical cells. Right, why is it important? So growth and repair. So we start out as one cell, we grow, and children grow into adults. Adults have more cells than children. That's because of mitosis. Right, so interphase was the first stage. So on these cells here, the cells that way you can kind of just see just a nucleus. You can't make out any chromosomes. That is in interphase. So in interphase, most cells are in interphase and they're just doing their normal jobs. So if this is a muscle cell, it's contracting. If it's a brain cell, it's thinking. And what also happens in interphase is the DNA replicates itself. So that's what it looks like on a diagram. But interphase is not on this diagram at the bottom. That's why I've just told you about it. So cells are normally in interphase, but when they start splitting, that's when mitosis happens. Right, so there's 4 stages. You need to remember them. So PMAT, the first one's prophase, then metaphase, anaphase, and telophase. So I'm not going to tell you the definitions of them. You can watch this video again and pause it if you want. But the definitions of them things are on the screen here. What happens after mitosis is, so mitosis is the splitting of the chromosomes, so how the chromosomes go into 2 new cells. The cell itself splitting, that's called cytokinesis. So basically the cell membrane is made between the 2 cells and the cells bud off and split. So that is an overview of mitosis. How can this come up in your tests or your exams? So you might be given a match the part of the cell cycle to a definition of what happens. So again, you can pause this video and have a go. This is also in the revision worksheet, which I'll put on Teams. You might also put these in the correct order. So I'm going to talk you through this question actually. So what I would say, if you've got a question like this, in the margin of your exam or your worksheet, write down 1, 2, 3, and 4. Now, which one of these do you know? So meta means middle. So which one of them are they lined up in the middle? Well, that's number 2. So metaphase. I would also be writing PMAT down there. So now we need to put them in order. Right, number 1, that's going to happen last. So the new membrane's grown around the chromosomes. Another thing I want to note here is that on number 2, the chromosomes are in the middle and they are X-shaped. So they're these are the double chromosomes. On number 3, they've split apart. So they are no longer the X double chromosomes. They've been split. So 3 must happen after 2 and 1 must happen after 2 because the chromosomes are being split apart. I hope that makes sense. But number 4, they are also X chromosomes. So that actually happens at the start. So number 4 is prophase. That happens first. And what's happening there is they've formed the X-shaped chromosomes and they're starting to make their way into the middle. Then what happens next is number 2. Then Once they've lined up in the middle, number 3 happens. They are pulled apart in anaphase. And then number 1, that's telophase, where they have reached the opposite ends of the cell. So the correct order is 4, 2, 3, 1. So the right answer must be 4, 2. So it must be C, must be the correct answer. So you could get questions like that. You could also get a microscopic image, and it might say explain which of these stages are which. So I just want to show you the stages, what they look like. Right, so I did say interphase, you just see one nucleus. Prophase is when the chromosomes start forming. So this here, that's in prophase. This one here is also in prophase. This one is in later prophase. So they've started to try and find the middle of the cell. So none of these are in metaphase, but what you would see is they've kind of all lined up in the middle. You would see a definite band in the middle. None of these are in metaphase. In anaphase, they start getting pulled apart. So this is kind of late anaphase before telophase happens. And then this one here is in late telophase. So you can see one cell with 2 nucleuses. So that's telophase. So you might be asked, this is more A-level questions, but you might get asked that if it's the really high end, like questions that are trying to stretch the level 9 students, okay, you might be asked what stage is this cell in. So that's mitosis. So it's for growth and repair. There's 4 stages to mitosis, but before that there's interphase, and after that there's cytokinesis. So you do need to remember them. It is really complicated, but the more complicated thing about it, I think, is the sheer number of keywords. So I think there's probably 10 good keywords that I've talked about there. Things like diploid, things like anaphase. There's tons of keywords, so make a glossary and make sure you understand them different keywords. Right, next thing in this topic is growth curves. So babies' weights, babies' lengths, things like that, they are expected to follow a growth curve. So what happens is things grow faster when they are younger than they do later on. So adults stop growing, so they wouldn't have a growth curve. That would be flat. So babies grow faster younger, so you have a growth curve. Right. 2 types of question I can see come up on this. So what does this mean? So a baby is in the 90th percentile for mass. What does this mean? Well, that means, so if a baby's in the 90th percentile, that means the baby is heavier than 90% of other babies. If it says a baby is in the 75th percentile for its length, its height, what does this mean? It is longer than 75% of babies of the same age. It could say like a baby's in the 10th percentile for its head circumference. What does this mean? The baby has a bigger head circumference than 10% of other babies its weight. So you just need to answer it like that. Other questions that could come up is things like this. So analyzing the graph, what is the value for the 50th percentile for a mass of a 6-month-old baby boy? So you would have to look up 6 months up to there till you get to the 50th percentile, and then you would move across. So this one here, so that would be 7.9 kilograms. How much should they increase? Something like that. So you would find the first value, and then you would find the second value, and you would take the small number away from the big number and find the increase. It could say as a percentage increase. 2 seconds. Someone's at the door. I will take 2 seconds. Brilliant stuff. Back. Sorry about that. Yeah, so you could also get percentage increase questions. So what is the percentage increase in mass between 2 months and 10 months? So that's when you do new take away old divided by old. Growth curves, that's it. That's all you really need to know about them. Right, another thing that comes up on this page in the textbook, and it's not really mentioned again, is the idea of differentiation, another keyword. So the embryo, the human zygote, that's made of one cell. When it's split to 8 cells, when it's split a ball of cells, them cells don't really look like anything special. But when development has started happening, babies have quite clearly got eyes and fingers and ears and brains. So the cells have started to turn into new types of cell. What this is called, that's what differentiation is. So when a cell turns into a specialized cell. So red blood cells are made from stem cells. The stem cells are not blood cells, but what happens is it splits into blood cells, and then the cell turns into a blood cell through this process called differentiation. So red blood cells have no nucleus, and they have a thing called hemoglobin on their surface, which attaches to oxygen. That makes that cell brilliant at its job. Nerve cells have a thing called a myelin sheath, which we're going to look at later. They've got the weird shapes. Muscle cells can contract. They've got proteins in that allows them to change shape, which allows your muscles to move your bones. So different cells have got different jobs, and they are good at the jobs because of this process of differentiation. Right, plant growth. Right, 2 ways that plants can grow. They can make more cells. So if they double the amount of cells and them cells are the same size, they're going to double their length. And also the cells that they have, the second way they can grow is their cells can elongate, so that this actual cells can get bigger. And when plants grow, they do both of them things. So that again, tons of keywords in this. So first thing, root cap. So this is the very tip of a root in a plant. These are microscopic, they're very, very small. The root cap, that's there for protection. And this is not splitting, it's not doing anything exciting. It's literally just there as a protection. It's like a little helmet on the end of the root, which is going to burrow through the soil. Right, the bit behind the root cap, that's the interesting bit. So it's called the zone, pardon me, the zone of cell division. And this is where the stem cells in the plant are. So plant stem cells are called meristems, and they are found in the tips of roots, and they are also found in the top of the shoot as well. So as the plant grows taller and also the roots grow longer, that's where they are found. Say again, sorry, Farbod.
What's the zone called again?
It's called the zone of division.
Okay.
So here, each cell is splitting into 2 new cells. How do you think, while I've got you on there, Farbod, how do you think them cells are dividing into 2 new cells?
Mitosis.
Brilliant. So that's a sort of question that could come up on your exam. How are the cells dividing there? It's mitosis. The cells are dividing. So each cell's turning into 2 new cells. Meristems are just plant stem cells. And so the cells there are splitting into 2 new cells. So this is the newest part of the plant. Like these cells are the youngest. So as we go up the root, the cells are getting older. And what you find is right at the zone of division there, the cells are tiny. But what happens here is behind that, the cells are a bit older. And what's happening is they are stretching out, they're elongating. So that's also pushing the zone of division further away. So the cells are splitting and also elongating. So that's how plants grow. Behind that is a zone of differentiation. So this is where differentiation is happening. So all of the cells are pretty much the same where the meristems are. But here they're turning into xylem. So a thin tube is forming there. So all these cells are kind of bunched together. But here they're forming a tube down the middle, which is kind of attached like one thin tube. That wasn't there before. So they are differentiating into xylem cells there. They're differentiating into root hair cells here. So they are turning into different cell types. And in the shoots, they'll be turning into xylem cells, but also palisade cells, which are there to capture sunlight. So differentiation, the cells are turning from one cell type and becoming the cell type that they will be for the rest of their life. Right, plant growth, done. Right, so stem cells in adults, there are two different types. They are embryonic stem cells, which are found in embryos, and embryonic stem cells can become any type of cell. So if you think about this, we started out as a zygote, and that has become all of our cells that we have as adults. So embryonic stem cells, these are found only up until the 8-cell stage. So the 8-cell stage, if you removed each of them 8 cells, they could all turn into copies of you. So you could have, so if you split up them 8 cells and 3 of them survived through pregnancy, you would have identical triplets. So the 8-cell stage is the last stage at which embryonic stem cells are found. Once the 16-cell stage happens, these start differentiating. So one of these might become, it might be able to make brain cells, but it might not be able to make heart cells. And then another one that might be able to make heart cells, but it won't be able to make bone cells. So they start differentiating only after the 16-cell stage. So embryonic stem cells are found before and up to the 8-cell stage. After that, you consider these as adult stem cells. Right, when it talks about where can you find adult stem cells, the best answer you can give is in bone marrow. So adult stem cells can't become every type of cell, but they can become a few different types of cell. So in your bone marrow, these can make all of the cells that are found in your blood. So for example, here we have red blood cells, white blood cells, which are to fight infection, platelets, which are there to heal cuts. So adult stem cells can become a few types of cells. Embryonic stem cells can become any type of cell. Right, halfway through already. Not bad. Right, so the brain. So again, the brain develops really early on in the embryo. And there are 4 bits of the brain that you need to know. So the spinal cord's not really part of the brain, but the brain and the spinal cord make up a thing called the central nervous system. So the CNS is made of the spinal cord and the brain. Right, if you move up from the spinal cord to the brain, it starts off more primitive and gets more evolved and more human as you go up. So the medulla oblongata, that controls things like your heart rate, breathing rate, swallowing food. These are all things that you do without consciously thinking about them. So this is sometimes called the lizard brain, things that like really primitive animals can do. So yeah, when you start exercising and your heart rate goes up, you don't think, oh, I'm going to make my heart beat faster because I'm doing exercise here. Your medulla oblongata just controls these things. Like, I've chewed my food, I'm going to swallow it now. You don't consciously think, I've chewed that food sufficiently, I'm going to swallow it. Your medulla oblongata just controls these really primitive sort of reflexes. Next bit, cerebellum, which is Latin, just means little brain. So it's kind of the shape of the cerebral cortex, and it's underneath it. This controls fine motor movement. So athletes, musicians have very well developed cerebellums. So yeah, piano players and stuff, their cerebellum is more developed than people that can't do these sort of things. So fine motor movement happens in the cerebellum. So questions you could get on this, it might be like, yeah, this tightrope walker, what would part of the brain would you find is more developed? Would be the cerebellum, because balance, fine motor movement happens there. Right, the cerebral cortex, this is the more human part of the brain. It's highly folded. It makes up 80% of the mass of the brain. And at the bottom here, you do not need to know any of these. You don't need to remember these, but there are different areas within the cerebral cortex which do different jobs. So the very front, things like emotions, how to learn, that's in there. Different like language and speech is in there. Different senses and tastes. So each area have got different functions. We'll come on to how we know this in a second, but that's the structure of the brain. So you do need to remember spinal cord, medulla oblongata, cerebellum, and then the cerebral cortex. You do need to know them, and you need to know their general jobs. So the cerebral cortex, if you had to give it one job, it would be thinking. So anything that you think about, so that tastes nice, or I don't know what that word Mr. Ingledew said means, that's all happening in your cerebral cortex. So its job is to think. Right, how do we know about parts of the brain? So I haven't put a picture in, but what's in the textbook is we've studied people that have brain damage. So there was a guy called Phineas Gage who basically had a spike speared through his brain, and he couldn't form new memories. So they figured out which part of his brain was damaged, and went, well, that part of the brain must be to make new memories. Two other ways that we look at the brain. So CT scans. A CT scan is basically a 3D X-ray. So on this picture here, this is the top of the brain, and then moving downwards through the brain, you get a 3D picture. So the CT scan shows structures within the brain. So any damage to the brain, and also it'll pick up things like tumors. So this person has a large tumor in their brain, and that is shown by a CT scan. Right. Another type of brain scan is a PET scan, which we did in physics, and you need to know the physicsy part of it for physics. You need to know the biology part for biology. So a PET scan shows how active a part of the brain is. So we'll do a little bit of physics, but basically you get a radioactive glucose. And what happens is when that glucose is used up in physics, you need to know that it basically makes two gamma rays, and then gamma rays are picked up by a camera. So if the person's head is where my pointer is, that gamma ray is going to get picked up on one side of the machine faster than it will be on the other side. So it basically, the machine puts it together. Where did that gamma ray come from? And what it does is, so because it's using radioactive glucose, which part of the brain is going to use more glucose? Well, it's going to be the more active part. So a PET scan shows brain activity, not just the structure, but how it's being used. So what you would do is you would get somebody. So if someone comes into the hospital with eye problems, they can't see properly, you're thinking, is it a problem with the eyes or is it a problem with the brain? So what you can do is you can do a PET scan, so you can give them some of this radioactive glucose, and you can make them look at stuff. And then you can analyze the PET scan. Did the part of the brain that should light up when they're looking at stuff light up? If it did, brilliant. It's not a brain problem. It might be something to do with the eye or the optic nerve. Did it not light up? Well, that means it's probably a problem with that part of the brain. So a PET scan shows activity, whereas a CT scan shows structures within the brain. And we can put that together. So this is how we know that there's visual centers in the brain and parts of the brain that understand speech and language, parts of the brain that light up when you taste something sour. So we know that because of PET scanning. Right. Oh, one more thing. So how, if someone has a brain tumor, what are the possible options? So number one, surgery. So you can physically open up the cranium and remove it through surgery. Another one, radiotherapy. So this is using gamma rays to target and to kill them cells that are making up the brain tumor. So again, we did this in physics. So you shoot the gamma rays from different angles to maximize the damage to the tumor, but minimize the damage to the healthy areas of the brain. So you shoot it from one angle, then another angle, and it focuses in on the tumor. So radiotherapy uses gamma rays to kill it. Chemotherapy uses chemicals which kill cancer cells more than they kill healthy cells. So chemo, chemical, radio, like the electromagnetic spectrum. Hopefully you'll remember radio waves, electromagnetic spectrum, gamma rays. So radiotherapy uses gamma rays, chemotherapy uses chemicals. Right. So questions you could potentially get in tests or exams. It might give you a map of the brain and say this lit up during a PET scan. What's happening? What's that person doing? So the back of the brain, well, you would get the mark if you said that person's seeing something because it's a vision area. The middle of the brain there, what's in the middle of the brain there, just above them to that lumpy bit there. Well, it's this bit, which is hearing. So they're probably hearing something in C. You could get things like that. You could get generally, what does the cerebellum do? Things like that.
So do you have to like remember all these like little functions, or do they give you?
No, so you are expected to remember the four parts of the brain, what a CT scan is, what a PET scan is, but you are not expected forward to remember that this is the emotions part of the brain. You would, if a question like this came up, you would be given a map of the brain. So you wouldn't get just like part D of the question there. What does that do? You wouldn't never remember that. But if you had a key as well, like a diagram which has this on, well, that must be the speech area. So this is lighting up so that person that's doing this is probably speaking. So no, you wouldn't be expected to remember the different parts of the cerebral cortex and what they do, but you might be asked to interpret what that's showing. Right, another thing, so another keyword was quadriplegia. So a quadriplegic is someone who has all four limbs, which cannot be used because they have a problem with their spinal cord. So this is Christopher Reeves, who played Superman, in the early Superman movies, and he fell off a horse. He damaged his spinal cord pretty high up, and all four of his limbs are disabled. So quadriplegics can't use all four of the limbs. Basically, if you damage the spinal cord, the higher it is, the more chance you have of serious damage. So if you damage it really high up, you can die. So this is a pretty gruesome thing about, but when they used to hang people, it snapped the neck really high up and they would die immediately if they were hung from a long drop, and it snapped their spinal cord high up, horrific to think about. If you damage your spinal cord further down, you could end up quadriplegic. If you damage it like towards the bottom of your back, you might be disabled from the waist down. And it's basically, so the nerves that go into your spinal cord generally go out horizontally. In general, you can find a diagram on Google if you want and just say like spinal cord nerves. You'll be able to have a look at where they end up. But the further down your spinal cord, wherever that's damaged, the further down, generally the less serious damage it does. I mean, it's pretty serious to be permanently disabled from the legs down. But yeah, so spinal cord damage was also on that part of the textbook. Right. The last general things about the nervous system, the eye, and then how nerves are organized in the body. Right, so the nervous system is made up of nerves, neurons. You need to know and remember all of these different parts of the neuron. So this bit here, I think it's very, very complicated. You're probably better off drawing a diagram of this, drawing it out and remembering the names. So we're going to start at this side because this is the side that the nerve impulse happens first. So this is called the dendrites. The dendrites pass the nerve impulse down this bit called the dendron. This is the cell body. And then this goes from the cell body to the axon and then the axon terminals. So how you're going to remember them. So it's actually backwards to alphabetical order. So you've got dendron, cell body, axon. So you can remember that as ACD, but it's like the other way around, isn't it? So it's DCA. So the dendrites happen first, then the dendron, cell body, axon, axon terminal. So that is a general structure of a neuron. The nucleus is in the cell body. Why are they specialized? Well, they pass on impulses. They're also very long cells. So compared to a red blood cell, which is like a disc shaped, these are really long and thin. Why are they long and thin? Because they need to connect parts of your body which are really far away. They need to connect the tips of your toes to your brain. So these are really long cells. I'll just point out there that the tips of your toes, between the tips of your toes and the brain, there's lots of different neurons which connect together. So this is the structure of them. And another thing I want to point out here is these orange bits on this diagram. This is called the myelin sheath. So at A level, you'll learn that these are actually made of cells called Schwann cells. Basically, they're just layers of fat at GCSE. You can just think of them as layers of fat which do not allow electricity to pass through. So if there's no myelin sheath, the nerve impulse would go all the way up the cell, like that, all the way up, and it would take quite a long time. The myelin sheath, what its job is it speeds up the nerve impulse. So what happens here is the nerve impulse gets to the myelin sheath and it jumps to the next bit and jumps to the next bit and it jumps, and it actually saves time. So it speeds up nerve impulses a lot. So again, this is level 8 and 9 stuff. But you might be asked, well, what's the difference between a myelinated axon and an unmyelinated axon? So a myelinated axon would have faster impulses than an unmyelinated axon. So some nerves in our body have the myelin sheath, and they are generally ones where you want a really quick nerve impulse. So, for example, your eyes, you want to see things really quickly. Your body wants to experience any senses really quickly. So all of your sensory neurons, which we'll get onto later, they are myelinated because you want them to speed up the information that's transferred to your brain. Right. This is, again, it's A-level this. I apologize, but I do feel like telling you these things will be worth it. Hopefully it makes a bit more sense. Right. If we think about the heart now, so you've got 4 chambers of your heart. We're going to do this in more detail in year 11, but the top of your heart contracts before the bottom of your heart, and you actually want a delay between the top of your heart contracting and the bottom of your heart. So the nerves that tell the bottom of the heart when the top of the heart has contracted, you want a bit of a delay. So you have unmyelinated axons, which take longer. So there's a slight delay there, and that's sometimes beneficial in the human body. So our heart has unmyelinated axons. Some animals, such as frogs, they haven't developed the myelin sheath, that they haven't evolved that. So some animals only have unmyelinated axons and they are slower. So yeah, that's a lot of information about the myelin sheath, but it hasn't been asked in exams, and I feel like it's due for you guys. I really do. Right, so dendrites, dendron, cell body, axon, axon terminal. That's what the nerves look like. And very, very quickly, nerves are made up throughout the body. So they connect sensory organs. So in your fingertips, in your retina, in your eye, in your ears, in the cochlea, all these things that sense different things, they are connected to your central nervous system by nerves. This is where they are. I'm going to get on to the reflex arc. That's going to be the last thing we do. But I just want to show you that we have, so these keywords, I just want to talk about these. Right, a stimulus. This is a change in anything outside of the body. So if you think about that, what's a stimulus? So a flash of light, that happens outside of the body. It's a change in the environment, is another way it's sometimes said. So that light wasn't there before, a flash of light, it is there now. So it's changing the environment. Something hot touching your skin, that is a change in the environment. So a stimulus is a change in the environment. A sensory organ is something that basically senses that change in the environment. So your retina in your eyes, the cochlea in your ears, you've got heat and touch receptors in your fingertips. They're all sensory organs. Right, sensory neurons connect the sensory organ to your central nervous system. What is the central nervous system? It's the spinal cord and the brain. The central nervous system decides what to do with this information coming in, and it passes it on to motor neurons, which connect the central nervous system to effectors. So again, we're going to talk about effectors in a bit more detail in a second, but effectors are either muscles or glands. And then that has a response. So the response is really bright light flashes, you blink. Something hot touches your finger, you pull your hand away. So these responses, this is how the nervous system is organized, and we'll talk about a few other things in a little bit more detail soon. Right, so the effectors, yeah, there can be muscles, and other ones are glands. So, right, what do we know? Well, adrenaline's made by the adrenal glands. So, a tiger bursts through your bedroom door now, you're going to release some adrenaline. So what's happened is you've seen the tiger, your eyes have sensed it, sends a message to your central nervous system, motor neurons send a message to your adrenal glands, and the response is you release some adrenaline into your bloodstream. That makes your blood pump, your heart beat faster, and you can run away quicker. So Effectors can also be glands, other things. So you eat some sugar, that is sensed. I can't even, oh my God, I can't remember. What senses blood sugar? Look it up. Is it the pituitary gland? I think it might be. Then your body decides what to do with that. It sends that message to your pancreas, which releases some insulin. So these stimulus response things aren't just seeing, touching. It can also be you get a bit hot, that's sensed, and then you sweat more. So it can be things like that. And we'll do a lot more of this in the hormones topic. So we will look at things like insulin. We've done, in the pituitary gland and the menstrual cycle and things like that. So we'll do a bit more of that later in the course. Right, the eye, very, very complicated organ. Lots of evangelical Christians use the eye as, well, God must exist because this could never happen because of evolution, because it's far too complicated. There's actually some really good YouTube videos which explain how the eye has evolved. So it did used to just be one light sensitive cell, but this is what it is, right? You need to know this. So we'll start at the front. So the cornea is basically just some see-through skin. It's there to protect the eye, but it also refracts light towards the lens. So any light coming in at the top is refracted towards the lens. Right, next bit, the pupil. So the pupil isn't actually a thing, it's actually an absence of a thing. So if you look in the mirror and you see your pupil, that's actually a hole into the back of your eye. The back of your eye is dark because there's not much light getting in there. So the pupil is actually just a hole, and we can prove that when we dissect eyes. The pupil is surrounded by the iris, which is a ring of muscle, and this is the colourful part of the eye. So if you've got blue eyes, this bit's blue. If you've got brown eyes, the iris is brown. And what it does is it regulates the amount of light entering the pupil. So if it's dark, what will happen is some muscles in the iris will contract and some will relax, and it will widen the pupil. If you are in like bright sunlight, the iris, the other parts of the iris will contract and the other parts will relax. It'll be the opposite. And what will happen is the pupil will get really small. So the iris regulates how much light's getting into the eye because you want a certain amount of light on the retina. Next bit, the lens. This focuses light on the retina. And if you've read stuff for a long period of time, you realize your eyes hurt. So the lens physically is changed shape by this thing called a ciliary muscle. And it's painful. So if you look at stuff really close up, it hurts your eyes. So these do use quite a lot of energy. And when things are really close up, this needs to be really fat, the lens. When things are really far away, the ciliary muscles contract and the lens becomes really thin. So the lens can change shape depending on how far away the thing that you are looking at is. And this makes a clear picture on the back of the eye. Right, the fovea is not actually in the GCSE topic, but this is where light wants to be focused on. So it's a part of the retina which has really, really good color vision and really clear vision. So basically the light is focused on there. The retina itself contains light sensitive receptors. When there is light, they give a signal. When there's no light, they don't give a signal. And our brain interprets that into vision. Last part of the eye is the optic nerve, which connects the eye to the brain. So it's basically connecting all the information from the retina, putting that together and giving that information to the brain. So the eye, very, very complicated. Again, how many keywords have we got there? Well, we've got like 8 new keywords there. I'll give you another one, number 9. So this white bit of the eye is called the sclera, so the bit surrounding it. And this is really, really tough, and it's for protection. So the eye, how are you going to learn that? You can make a glossary. You can do thingy cards, what they're called. You can write these down and just what is the definition of the lens, write the definition on the back, or what's the job of the lens, what's the function of the lens. It's the focus light, things like that. So yeah, you will have to remember all them parts of the eye. Enjoy. Right, eye problems. So the first thing is long and short-sightedness. Right, short-sightedness is when the light converges too short. So this is caused when your eyeball is too long, or it could be a problem with the front of your eye as well. It's converging light too much. So how are we going to fix that? Well, we need to spread the light out a bit more so it actually focuses on the fovea on the back of the retina. So the correction for short-sightedness is a diverging lens, something that's going to spread the light out slightly so the light focuses on the retina. Right, next thing. Long-sightedness is when your eyeball is too long and the light isn't focusing enough. It's not converging enough. It's converging behind where the retina is. So to fix that, you have a convergent lens. So again, I could have put this into a table for you, but you need to think about how you're going to remember these fairly difficult things. So I would maybe make a table out of this information so you remember it. So problem, short-sightedness. What's it caused by? Eyeballs too long. How are you going to fix that? Divergent lens. Problem, long-sightedness. Caused by eyeball too short. A correction with a convergent lens. Again, so many keywords. Convergent lenses and divergent lenses, these come up in physics as well. Yeah, enjoy all these keywords. I think that is the toughest thing about biology. But yeah, the first problem is long-sightedness and short-sightedness. Other problem is color blindness. So this is the colors that normal people can see. If you have, so we have 3 color receptors in our retinas if we're healthy. One senses blue, one senses red, one senses green. If your green color receptors are faulty, you can still see blue and you can still see red and you can still see between blue and red, but you can't see green. So you can't tell the difference between blue and green or red and green. So yeah, these people that have damage in the green receptors can kind of still see like blue, red and purple, but they can't see yellow. They can't see like turquoisey colors. Anything with green in, they can't see green itself. So color blindness is caused by one of the types of cells in your eye being damaged. So
you know the
yep.
So you know the colorblind glasses. What did they do?
The what? Their new glasses.
The colorblind glasses.
Yeah. I've seen YouTube videos of people wearing them, but I've actually got no idea. I'll tell you what I'm going to do. I'm going to watch. I'm going to find out, and I'm going to tell you how they work. Hopefully, yeah. But I really don't. I've got no idea. But they're not attached to the brain, so it can't be like a sensor. It must do something with the wavelength, and I don't know. Okay. I really don't know. I'll find that out. That's a great question. But yeah, so in our retina, sorry, I will point out there's 2 different types of cells. There's rod cells, which basically see in black and white, and there are cone cells which see in colour. And we've got 3 different types of cone cells which see blue, red, green. And you can have a problem with one of them cone cells. If you've got problems with your rod cells, you don't see very well. So there's different types of colour blindness. It's more common in males. It's an X-linked trait. Hopefully you remember what that is from genetics. So males are more likely to get it because females in general will have, if they've got one faulty copy of the X chromosome, they can have a healthy copy. They get a healthy copy as well. Males, if their mother has one faulty copy, she won't be colorblind. But if the male has a 50% chance of getting that faulty X chromosome and getting it, so males are more likely to be colorblind than females. That's certainly true in some types of colorblindness, but the more common types, males are more likely to get it. Right, and then the last one is cataract. So this is when a cloudy protein builds up in the lens and then the jelly part of your eye, and it can cause blindness. It's pretty easy to treat in the Western world and the developed world, but it's a real problem in the developing world. So cataracts. Right, the last thing. Any questions so far, by the way? All right. Right. So synapses and reflex arcs. So a synapse is simply the gap between 2 nerve cells. So we've got like this could be a sensory neuron then, and then a relay neuron. Anything between 2 nerve cells is called a synapse. Right, this is an A-level complicated diagram. I'm just showing you, if you do A-level biology, this is the sort of stuff that you're going to need to learn. The only thing I want to point out here is, so the nerve impulse comes to the end of the dendron, sorry, on the end of the axon terminal. So the nerve impulse reaches the axon terminal. Then that nerve releases a thing called a neurotransmitter, which transmits the information across the synapse. Then this is picked up by the dendrite. It senses that neurotransmitter and passes on a new nerve impulse. So we'll do that again because I messed up the first time. Axon first, the nerve impulse comes, it releases some neurotransmitter which goes across the synapse, which is sensed by the dendrite, which passes on a new nerve impulse. So synapses are really, really important because they mean that nerve impulses can't go the other way. What could get asked in your exams, I've just thought of this now, is like differences between nerves and electrical circuits. So in the electrical circuits, the electricity, the current can go the other way. Nerve impulses can only go one way, and that is largely due to the synapse. So there's parts of the synapse which have these, which can release neurotransmitters, and one side which can sense neurotransmitters. The dendron can't release neurotransmitters, and the axon can't sense neurotransmitters, so that it can't work the other way around. So synapses make nerve impulses go in one direction. What else can they do, which is pretty cool? Well, one nerve can send a message to 2 new nerves, so it can split nerve impulses up, and it can also bring them together. So in your optic nerve, you haven't got as many nerve cells as you have light sensitive cells in your retina. So you'll have millions of light sensitive cells in your retina, but you won't have millions of neurons. So what's happening there is the retina is building a picture. It's putting that information together into some nerve cells, and they are sending that to the brain. So synapses, 2 points, they make it go in one direction, and also they can either split up or bring together nerve impulses. Right, I think 2 more slides. Right, different types of nerve cells. So they are the sensory neurons, which are attached to receptor cells. So this could be the retina. This could be cells that sense smell in your nose. Anything that's attached to a receptor cell, that's called a sensory neuron. Right, next, you have relay neurons. So these are found in the spinal cord, and you've got them in your brain cells, in your brain, sorry. What they do is they connect sensory neurons to motor neurons. So they figure out where that information should go. Then your motor neurons, these are connected to either muscles or glands. So they get a nerve impulse and either make a muscle contract or a gland release a hormone. So the 3 different types of neurons, fairly complicated. They've got kind of different shapes. You might be asked which one's which. Generally, if it's like really like fairly round looking, it's going to be a relay neuron. If it's connected to a muscle, it's going to be a motor neuron. If it's connected to the retina, it's going to be a sensory neuron. And I think this might be the last thing. This order again. So stimulus, sensory organ, sensory neuron, central nervous system, motor neuron, effector response. You need to remember that, and you need to be able to think of examples. So I'm going to give you 2 examples here. Stimulus, so you hear a loud bang. Your ear figures out, it senses that. The loud bang's an external stimulus. It comes into your body through your ears. The auditory nerve, don't need to know that. It's a sensory neuron. A sensory neuron passes that to the central nervous system, passes that to a motor neuron, passes that to an effector. In this case, it might be the muscles. I don't know what happens when you hear a loud bang. You jump. So your muscles make you jump. That evolutionary might make you, like, if it's a bang, it might make you want to get away from it. So that jump might make you survive more. Another example, so you see a bee or an insect flying towards your eye. What senses that? Well, your eye sees it. The retina detects that light. That passes it through the optic nerve to your central nervous system, which passes it back to a motor neuron, the muscles in your eyelid, and you would blink. So these are reflex arcs. These are things that you don't think about, things that happen instinctively. So something hot touches your finger, it's sensed by heat receptors in your finger. That's passed through a sensory neuron to your central nervous system, goes through a relay neuron, passed to a motor neuron to the effector, which is a muscle in your arm. That contracts, and the response is you pull your hand away. So reflex arcs happen without thinking about them, and we do these, the things that help us survive. So help us not burn my finger off, help us not get a damaged eye because of a bee. And these are different from conscious actions. So what happens is things like when you're hearing some information, your ear sends that to the central nervous system, which sends it to your brain. Your brain thinks about what to do with that information, and it might store it. So this is different to thinking. Reflex arcs happen just without thinking about it, yeah, unconscious. And that is B2, cells and control. Anyone got any questions about anything? Get in. Right, what I will say is I'm going to put this, put the revision question booklets on Teams. And I'll also put the end of topic test that you've done already, but you haven't seen for a while, also on Teams and the mark scheme. Make sure you have a look at them. So listening to me talk is not going to make you learn. What's going to make you learn is figuring out, do you understand this stuff and can you apply it to exam questions? I hope that makes sense. But you basically, to get better at science, you don't just need to listen to a teacher. You need to try questions. You need to figure out which questions you get wrong and obviously, if you're getting them questions wrong, you don't understand that thing in the right way. So anything you're not understanding,